All right, so welcome everybody. This is going to be the fourth and final lecture for our modern molecular biology course this year. And we're have as our teacher today, Martha Zuniga, who also participated in this course last year. And if you, a number of you I know were here, and Martha did such a great job last year. We're really delighted to have her back again this year. So Martha and Rusty and I actually go back way before Santa Cruz. Rusty and I were postdocs at Yale University in the late 70s, and Rusty was in the biology department and Klein Biology Tower, 9th floor, and there was Martha Zuniga, okay, working on her PhD. So Martha was born in Laredo, Texas, one of 10 children. She got a scholarship and went off to eventually get a BA degree from the University of Texas in Austin. And then she went to Yale, where she got her PhD. She did a postdoc in Caltech, so that was coming out to California, a very eminent immunologist, Lee Hood at Caltech. And they got a faculty position back at Texas. But we wanted to get Martha here in California in Santa Cruz. And so we hired her away from Texas in 1990, so 30 plus years for Martha at Santa Cruz. She's an immunologist, been teaching immunology in the research lab up at the university. And so today she's going to talk about one tiny bit of a very complicated field of immunology. So Martha, it's all yours.
Thank you, Barry. I'm going to share my screen. Now, I know everybody's muted, but I hope that people will interrupt me with questions. If I'm in the presentation mode, I can't see questions in the chat. So please just say something. Yeah. So the second half of this title is really appropriate. The crazy way that the vertebrate immune system deals with an ever-expanding antigenic universe. Oops, I'm trying to forward my slides here. Okay, so the challenge that the immune system has is that it must defend against all foreign molecules and organisms, even those that haven't yet evolved. We're familiar with the emergence of new pathogens, such as SARS-CoV-2, which has put us in lockdown for a year and a half. And so I want to talk today about, a lot of my presentation is going to be background. My actual research is going to be a tiny bit of it. And I chose to do that because if you don't understand all of the background, then my research won't make any sense to you. So the vertebrate immune system consists of two separate systems that interact with each other. The innate immune system shown here on the left and the adaptive immune system. There's a few cell types that have properties of both. I'm not going to talk about those. Today, I'm chiefly going to talk about T cells, which are over here on the right. And we're specifically interested in CD8 positive T cells, which kill virally infected cells. So just a little bit of nomenclature here. The cells of the adaptive immune system are called lymphocytes, but we immunologists interchangeably use B cell for B lymphocyte and T cell for T lymphocyte. It's just fewer syllables to say B cell versus B lymphocyte, for example. So the lymphocytes are the workhorses of the adaptive immune system. They're found in the blood, in lymphoid tissues, and in most organs of the body. And they express antigen receptors that are specific for molecular entities, antigens, and mediate antigen-specific immune responses. So I'm just going to walk you quickly through the adaptive immune response. The first time you get exposed to an antigen, either by infection or by immunization, it gets engulfed by a specific cell of the innate immune system called a dendritic cell. It carries it from wherever you got exposed to the antigen. So for those of us who got the SARS-CoV-2 vaccine, it was in our arm. It'll go to the most nearby lymph nodes where it can present the antigen to a type of T cell called a helper T cell. And it can stimulate this T cell to then proliferate and differentiate either into a helper T cell that can mount an immune response or to a memory helper T cell. At the same time, the antigen is presented to another cell type, a B lymphocyte or B cell. But the interesting thing is that this B cell cannot continue its progression to develop into a plasma cell that secretes antibodies or into a memory B cell unless it also gets growth factors called cytokines from the helper T cell. At the same time, the antigen that is taken into the lymph nodes by dendritic cells is presented to a CD8 positive, I'll come back to this nomenclature in a minute, or cytotoxic T cells. This is the type of cell that once it differentiates and proliferates can become an active cytotoxic T cell that can kill virally infected cells, or a memory T cell. And the reason this is also beautiful is that these memory cells can protect us when we get a second antigenic stimulation. It's the memory B cells, the memory helper T cells, and the memory cytotoxic T cells that we're counting on when we get a SARS-CoV-2 vaccine or any other kind of vaccine. And although I don't study helper T cells, I want to point out to you that the reason AIDS, HIV, which leads to AIDS if not treated, is so devastating is that it's the helper T cells that get infected by the HIV virus. And if you don't have these guys, you don't have the necessary cytokines to stimulate these B cells and these T cells to do their job. Okay, but we're going to focus on the development of T cells, and I'm going to talk a little bit about the cytotoxic T cells. So there's 4 related but distinct cell membrane proteins that are responsible for recognition of antigen by cells of the immune system. On B cells, it's membrane-bound antibody or immunoglobulin. On T cells, it's T cell receptor for antigen, but they recognize antigen together with another kind of molecule called MHC, which we study in my lab, and I will talk more about today. So here are just little cartoons showing that the B cells with the antibody on its cell surface receptor, the T cells, T helper and cytotoxic with their T cell receptors. These 2 classes of T cells can be distinguished from each other by co-receptors called CD4 on the helper T cells and CD8 on the cytotoxic T cells. And we're going to be talking about these 2 molecules and their role in the development of T cells today. So, okay, one thing that was very puzzling to immunologists for the first 60 or so years of the 20th century was how is it that we can have B cells and T cells that are specific for any antigen in the antigenic universe? How does that happen? There were 2 schools of thought. One said, oh, there's a lot of DNA in the human genome. There's a separate gene for every single antigen receptor. And then there was another school of thought that said, that can't be. The antigenic universe is vast. The genome isn't vast. You can't possibly have a receptor for every single antigen in the antigenic universe. A great immunologist who now does neurobiology, not immunology, by the name of Susumu Tonegawa, did some critical experiments that showed that both schools of thought were right. So I'm showing you here just a cartoon of the 2 chains of the T cell receptor, the alpha chain and the beta chain. And in red here, we have the variable region gene segments. There's between 70 and 80. Interestingly, there's heterogeneity there. Some people have more, some have fewer. For the alpha chain variable region, 52 for the beta. There's another variable region gene segment called J alpha for the alpha chain and J beta for the beta chain. And then the beta chain has 2 more gene segments that generate new antigen receptors called D for diversity. So the generation of an antibody molecule or of a T cell receptor relies on the random gene rearrangement of AJ alpha with a V alpha and AV beta with AD beta and AJ beta. And then there's also what is referred to as junctional diversity at the joining parts of V alpha to J alpha or V beta to D beta to J beta. And this is how you generate this huge amount of diversity so that you can respond to basically any antigen, even one that hasn't evolved yet.
Martha, I've never fully understood this. How big are these pieces?
Oh, that's a great question. So the variable region gene segments encode about 100 amino acids. The J's encode only 13 amino acids, and the D's encode only 3. So cloning the D's was actually, or identifying the D's was actually quite difficult. Lee Hood's lab did that first. And then there's also this junctional diversity. So, and then you can combine different, oops, sorry, I didn't mean to do that. Different alpha chains with different beta chains. So you can just generate a massive amount of diversity. I'm going to show you a figure. If you go ahead.
So this is kind of a random shuffling of the genes so that you can get a whole variety of combinations of amino acids to make random proteins, essentially.
Yes.
Okay.
Now, the C here stands for constant. So it's only it's about half of the alpha chain or half of the beta chain that has all this variation. The constant regions are called constant for a reason. They are constant. And I'm going to show you a diagram of the T cell receptor in a minute, so it'll make more sense to you. Thank you for those questions. So the T cells don't bind antigen alone as B cells do. The T cells bind an antigenic peptide derived from a protein, for example, a viral antigen, that sits in the peptide binding cleft of a molecule called MHC. And I'm going to talk about MHC in a few minutes. So the alpha beta T cell receptor has sequences that can interact both with regions of the MHC molecule and with the antigenic peptide. And here on the right, I'm showing you a ribbon diagram from X-ray crystallography. So the constant regions for beta and alpha are down here. The variable regions are here. And the really cool thing is these loops that contact MHC and peptide are the sequences that are brought together by the joining of V alpha to J alpha and of V beta to D beta to J beta. So the junctions, these sequences that are variable are exclusively in these loops that can interact with MHC and antigenic peptide. In fact, the variable regions themselves, the vast majority of them are not highly variable. The variable is in these loops, which is amazing. So the vast majority of the T cell receptor is a scaffold on which these variable loops are held up. Does that make sense? Okay. So I want to talk about MHC. I wish I had the time to give you the fascinating history of the discovery of MHC because I love it so much, but there isn't time. I'll just mention this great man, Peter Gore. He was a German cancer biologist. He was very interested in identifying tumor antigens, and he transplanted tumors. This was before inbred strains of mice were available. He actually captured mice from buildings in different neighborhoods of Berlin and isolated tumors from them, and then transplanted the tumors onto mice that he captured in the same building or onto mice that were from a distant part of Berlin. And what he found was that if he transplanted the tumor onto a mouse that was theoretically genetically identical or similar, the tumor grew and would eventually kill the mouse. But if he transplanted it onto a mouse that was from a different area of Berlin, the tumor didn't grow. This was in 1936. He published his paper. A wonderful immunogeneticist in the United States named George Snell actually had access to inbred strains of mice, and he wrote to Peter Gore and said, come to America, we can do this better with inbred strains of mice. You don't have to be capturing mice from tenement buildings in Berlin. But Peter Gore here had to flee Germany, and he fled to England, where London was being bombed by the Luftwaffe. I mentioned that because he thought he had a tumor antigen. He thought he had something that the immune system was recognizing because it was on a tumor. But at the same time that right after he published his studies in Germany, I'm sorry, in England, in London, these bombs were dropped on London, and people, innocent people in their homes, would be injured or killed. Some of them ended up with severe burns on their bodies, and they needed skin transplants to survive. And surgeons would take skin from healthy donors and transplant them onto the burn victims. And much to their horror, these skin grafts were rejected. And if they go back to the same donor and put skin again on that same recipient, it would be rejected even faster. They didn't put two and two together. They didn't realize that this was an immune response. But I just want you to know that they inadvertently discovered something that Peter Gore had discovered. And then Peter Gore, when the war ended, came to the United States, worked with George Snell, and much to his huge disappointment, these tumors were not being rejected because of tumor antigens. They were being rejected because of these major histocompatibility complex molecules. Because the name is so huge, major histocompatibility complex, we refer to them as MHC. He didn't discover two antigens. He actually discovered something far more important. He discovered how T cells recognize the immune system. So here's a ribbon diagram of a class 1 MHC molecule. There's two kinds, class 1 and class 2. My husband took a look at this ribbon diagram and he said, Bullwinkle the Moose. So here's Bullwinkle's snout, the beta-2 microglobulin light chain. His ears and antlers are made up of the so-called alpha-1 and alpha-2 domains of the molecule, and his neck is made up of the alpha-3 domain. And this portion up here, if you view it from above, as shown here, you can see that there's this nice cleft in which antigenic peptides can bind. And then the image on the right shows that we are hugely polymorphic for MHC. There's numerous variants in the human species. This is old data. I suspect these numbers are larger. These are the number of different variants or alleles for the HLA-B class 1 molecule, for the HLA-C and the HLA-A. So there's 3 genes in humans for class 1, and this gives you the number of variants that there are for each. Now, what is shown here in the middle, the red portions are where the variation in these alleles exists. They exist in the peptide binding site and in the sequences that contact the T cell receptor. So they exist up here. The rest of this molecule is relatively unvariable. So to recap, the T cell receptors and the antibody molecules generate diversity by combining all these different gene segments. The MHC molecules do it through polymorphism. And although I don't have time to talk about it, it's really fascinating that our breeding behavior actually predisposes us to want to mate with somebody who's polymorphic, who is different from us at these MHC molecules. That's a separate fun lecture that I don't have time to talk about today.
So Martha, maybe I'm asking a question on that directly. So when people do organ transplants or whatever, and you're doing tissue matching, is this what they're trying to match?
Exactly.
Yeah.
It's the MHC molecules. Both class 1 and class 2 have to be fully matched. If you're not fully matched, then you have to be given immunosuppressive drugs.
And what's the chance that one person will be a match to another person? It looks like it's a 1 in 1,000 or more.
You're really lucky if you have an identical twin.
Yeah. I remember reading about sexual reproduction and how it seems to be maintained partly because of defense against parasites.
Well,
and this looks like something that would make it really neat to be able to shuffle your alleles between generations so your parasites can't follow you or something. Anyway,
yeah.
it seems like it might be related, I guess is what I'm getting at.
It is. I don't think it's just parasites. I mean, pathogens general. And this is why it was bad for the royal families of Europe to constantly be marrying each other's cousins and whatnot. You really want diversity at the MHC. By the way, this is also a serious problem with bottlenecks that are created when a species is brought close to extinction. So if the San Diego Zoo has a panda and the Beijing Zoo has a panda, but they originally came from not that many generations ago from the same pandas, then you're going to have much less diverse. Oops, sorry. I keep accidentally touching this. Then you're going to have much less diversity at the antigen binding sites, and you're more likely to succumb to disease. So here's one thing you want to keep in mind. The polymorphism in the MHC, it's not just good for the individual, it's good for the survival of the species. Because, for example, going to our new enemy, SARS-CoV-2, even if there's people who cannot defend themselves, don't have the right T-cell receptor combination, variable region genes, same thing for B cells, same thing for MHC, and those individuals die, as long as the species has individuals that do have the capability of responding, the species will make it, right? So now I want to talk about how I've told you that the T-cell gene rearrangement events are more or less random. And the problem with that randomness is that they might generate T-cell receptors that are reactive against self. And it's essential that there's a mechanism for selecting the T cells that have useful TCRs. TCR stands for T-cell receptor. Useful TCRs are those that can bind self-MHC with foreign peptide, but do not bind too well to self-MHC and self-peptide. And all of these events occur in an organ called the thymus that sits right on the heart. So we generate new B cells and T cells every day of our life in bone marrow. There's stem cells that give rise to B cell precursors and T cell precursors. The T cell precursor stem cells go into the thymus, and that's where the T cells finish their development. We don't have time to talk about it today, but the B cells do all of their development in the bone marrow. And then at the end of this process, which is happening to all of us as we're sitting here, is that these B cells and T cells go to our lymph nodes and spleen, where they can then encounter foreign antigen. So the immune system is profoundly wasteful. It's generating all of these cells. It's getting rid of those that are autoreactive, which occur at high frequency because of the gene rearrangement events. And it keeps shipping them out to the periphery. And then if they don't encounter their antigen in a timely manner, they die. And so you're constantly replacing B cells and T cells every day of your life. Although the older you get, the less robust that process is. So I told you before, there's two major kinds of T cells, helper T cells, which express the CD4 molecule, which binds to class 2 MHC, and cytotoxic T cells, which express CD8, which binds to class 1 MHC. So these two cell types differ both in function. These are the ones that produce the cytokines that are necessary for the function of the immune system. And these are the ones that can kill virally infected cells as well as tumor cells. Okay. So the interesting thing is that these CD4 positive and CD8 positive T cells I just told you about, the helper T cells and the cytotoxic T cells, which each uniquely express only CD4, CD8, they develop from a precursor that expresses both. And I'm going to introduce you to a little bit of nomenclature here. So if the cells express both CD4 and CD8, we call those double positive. If they express only one, like CD4 only, it's called a CD4 single positive. And if it expresses, that's here, and if it expresses only CD8, it's CD8 single positive. So the interesting thing is that during their development in the thymus, two things are happening. They're being selected for being able to bind to antigen, and they're also depending on what their T cell receptor binds, they're deciding, I'm becoming a helper and I'm CD4 positive, or I'm becoming a cytotoxic T cell and I'm CD8 positive. All of this is happening at the same time in the thymus, which is amazing. So how does the thymus select for cells that can recognize? I mean, it's a crazy idea. And if you submitted this as a project as an undergraduate or a graduate student to your engineering professor, he would fail you, because you're randomly, it just seems like a system that shouldn't work. You're randomly generating T cell receptors, but you've got to get rid of the ones that are autoreactive. And you do that by selecting T cells that bind what I call underachiever C students, the ones that bind all right, but not great. And I'm going to walk you through that. So the T cell precursor rearranges its T cell receptor genes. This immature thymus site is expressing the T cell receptor and both CD8 and CD4. The first thing that happens to it is it undergoes what is called positive selection. It has to be able to bind to self-MHC. If its antigen receptor can't bind to self-MHC, it dies by what's called neglect. It doesn't get growth factors that it needs to survive. The ones that can bind self-MHC get those growth factors and they survive. The ones that don't die. They don't get the cytokines and they die. So you lose, okay, I'll tell you how many cells you lose after I talk about negative selection. So the first step, you're just asking, can you bind to self-MHC and peptide? You're not asking how well you bind. That's positive selection. The next step is negative selection. If the T cell binds to self-MHC, if its receptor binds to self-MHC and self-peptide too well, it dies. And so I'm fond of telling my students that the T cells must meet two seemingly contradictory requirements. They have to bind to self-MHC and self-peptide to survive. A students, B students, and C students. By that I mean T cells that bind super well, very well, or just moderately well, all survive positive selection. But in the next step, the T cells that bind too avidly die. So the A students fail. The thymus selects for underachievers. I can't tell you how much my students love this. Until I tell them that I don't select for underachievers. So the T cells that survive both positive and negative selection then emigrate to the peripheral lymphoid tissues. And just to show you how wasteful this process is, it is estimated that between 95 and 99% of the thymocytes survive both these processes. They either fail because they don't functionally rearrange a T cell receptor and they die. They fail because they didn't bind self-MHC at all, or they fail because they bind self-MHC and self-peptide too well. So going back to what I said before, an engineering professor would fail you on this because it seems like such a cockamamie way of doing things. But that's how the vertebrate immune system does it. So there's a sweet spot of affinity of the T cell receptor for binding to antigen. If it doesn't bind well enough, you have death by neglect. If you bind reasonably well, you generate mature T cells. If you bind too well, you die. Now, these selection events occur in specific areas of the thymus. This is a section of a thymus. And in the pale purple, we have a region that's referred to as the medulla. And then the darker purple is referred to as the cortex. Positive selection occurs in the cortex. It used to be thought that negative selection occurred in the medulla, but my lab and other labs indicate that it's more likely to be occurring at the junction between the cortex and the medulla, right here. So positive and negative selection seem to be separated both temporally. Positive selection happens first, then negative selection, and also are separated geographically in the cortex. The thymocytes are moving around in the thymus. They can move around in the cortex and in the medulla. And what you have in the cortex and the medulla that mediate the positive and negative selection are specialized epithelial cells. The ones in the cortex are referred to as cortical epithelial cells. The ones in the medulla are referred to as medullary epithelial cells. They express MHC molecules, but they seem to express different amounts of MHC molecules, and they also express different antigenic peptides. So if all of this worked perfectly well, then we would never have autoimmune disease, because all of the cells that bound to self-MHC and self-peptide 2 well would die. And you'd only have cells that could bind to self-MHC with self-peptide not well, with the hope that it's going to bind that self-MHC with a foreign peptide better. Now, this is the other thing that your engineering professor wouldn't like. This process of positive and negative selection doesn't guarantee that the T cell receptor will bind self-MHC with a foreign peptide better, right? There's no guarantee that that will happen. You really talk about a Hail Mary pass. There are several Hail Mary passes in the development of thymocytes. But autoimmune disease is all too prevalent. And why is that? There's several reasons. I'm only listing 2 here. First of all, not all self-peptides are present in the thymus, or they're not present at sufficiently high concentrations to be presented by the MHC molecules to the developing T cells. Also, the generation of the T cell receptor is random. The variable region generation is random, and the TCRs might not be able to bind sufficiently well to be deleted during negative selection. I want to point out that here, this region is what we're worried about, that maybe the affinity isn't so high that you die by neglect, that you manage to survive. So there's one more thing that I need to mention. We used to think that all we had to worry about was positive selection and negative selection, but more recently, several studies have shown that if the T cell receptor binds to self-MHC and self-peptide super well, not only does it not get negatively selected, it becomes a specialized kind of cell called an intraepithelial lymphocyte, or IEL, and these go to the gut, to the intestine. And so in my lab, we wanted to develop a mouse transgenic model that would allow us to study both negative selection and IEL selection. And I'm going to spend the next several minutes describing to you how those mice were developed. But before we do that, I want to make sure that there's no questions. And if there are, please ask them.
I have a question. I remember reading somewhere that the thymus, as you age, kind of fades away.
Yes.
And I was just wondering, you know, if we know what effects that has on the immune system as you get aged.
All right. All right. It's worse than as you age, Mark. Puberty is when your thymus starts to involute. By the time you are in your 6th or 7th generation of life, your thymus is hard to find because it's so seriously involuted. And the cause of this is the sex steroid hormones. The sex steroid hormones demolish the thymus, but not universally. So to answer your question, it seems that the major thing that's failing in that thymus as you get older is negative selection. So the positive selection still happens well, but for some reason, the negative selection events are not so robust. And this might be one reason that autoimmune diseases tend to occur later in life, that the negative selection is not happening as robustly as it should. But because positive selection is still happening, you can still generate T cells that can mount an effective immune response against foreign antigens. Now, why this immune system evolved this way? Who knows? I mean, that seems pretty crazy. But yes,
So I have a question. So since we have these two or three organs in our body that are essential to work or not work properly, I'm sure, has there been a study from the other side is how to keep them healthy? In other words, what we eat, what we do, so forth and so on. Is there an area of science that's looking at that?
Yes and no. I mean, there is a lot of interest in how nutrition and nutritional components actually boost immune responses, but the thymus involution, that is a hard nut to crack. It's not trivial to take a thymus from a young person and put it into an older person. And so there's not, I'm not aware of any way we could get around that problem. What has been done is the opposite. There have been situations in which I don't know who was brave enough to do, what surgeon was brave enough to do this, but I've read of situations in which thymectomies have been done on people so that you wouldn't generate more autoreactive cells. You wouldn't generate cells that are passing positive selection. That strikes me as dangerous because you could have a new pathogen emerging. I'm sorry, I want my immune system to still be functional. Okay.
Thank you.
Sure. Any other questions? All right. So I want to talk about our mouse model. And to do that, I'm going to have to tell you about some things that we didn't do, but that are essential for understanding. So do you know what transgenic mice are? I should have brought a slide on transgenic mice.
It's worth explaining.
Okay. I do have a slide on it, but it might take me a few minutes to fish it out, so I'll explain it without a slide. The generation of transgenic mice relies very, very much on some peculiar reproductive physiology characteristics of the mouse. One thing that's interesting is that when a female mouse is mated, her endocrine system produces hormones in such a way that her uterine horns are receptive to carrying a pregnancy. So if you mate a vasectomized male to a fertile female, she will, there's a window in time in which if you now implant fertilized eggs, she can carry them to term. A second interesting feature is that when a male mouse mates a female, he leaves a vaginal plug. And so you can, and this is critical for generating transgenic mice because of that window in time I just mentioned. So you can you'll have four kinds of mice. You will have vasectomized males and females that you superovulate with hormones so that they generate a lot of eggs. You also have a fertile female and a fertile male. Okay, so you take the fertile male and you mate him to the superovulated female. You look for that vaginal plug every 12 hours or so. When you know that the vaginal plug is there, that's when you want to harvest those eggs, because you have to harvest them when it's still a single cell stage. In the meantime, you've taken the vasectomized male and mated him to a fertile female so that you have a surrogate mother. Everybody always asks me, why do you need the second pairing? Why can't you just put the eggs back into the superovulated female? And that's because animal use regulations don't allow you to do it. Completely separate conversation about what they decide is good to do or bad, good or bad, but that's why you have to have two pairings. So you now take your fertilized eggs from the superovulated female who was mated with a fertile male, and you inject the gene of interest into those fertilized eggs. And now you implant those eggs into the female who is pseudo-pregnant because she was mated to a vasectomized male. And her body will be able to carry those fertilized eggs to term. You'll get baby mice out of it. So that's how you generate transgenic mice. And then you have to breed the progeny and make sure that they've got the transgene. So did that make sense to you? What would you like me to explain better? Can you put on your microphone?
Could you explain what the term transgenic means?
Transgenic, that's a great question. Transgenic means you're transferring in a gene. So the transgenic mouse is a mouse that has had a gene or set of genes transferred into its genome through the process that I just described. Any other questions? You're laughing. Is that because I didn't explain it clearly enough?
Do they tend to use CRISPR for this these days?
We can use CRISPR now. That's true, and that's a better way to do it. But I generated my mice without CRISPR because I did it so long ago. And yeah, right.
Okay.
So a group in a team of scientists in France so I forgot to mention, remember I told you earlier about MHC being highly polymorphic. In the mouse, the MHC, in the humans, I told you the class 1 MHC genes are called HLA-A, HLA-B, and HLA-C. In the mouse, they've got different names. And one of them, the ones that we study, is called H2K, capital K. And the different alleles have lowercase letters. So this CBA mouse has a different allele at K. It doesn't have the KB allele. It's actually got the KK allele, but I almost hate to tell you that because it might confuse you. So this mouse, the C57 Black 6 mouse and the CBA mouse, express different alleles at the H2K locus. This French group took cells from a C57 mouse, which expresses H2KB, and then immunized a CBA mouse, which doesn't have H2KB. And so H2KB was foreign, and then they cloned out T cells from this mouse, and they characterized them, and they selected for clones of T cells that had a T cell receptor that was specific for H2KB. And those T cells were CD8 positive and could kill C57 cells. Then they molecularly cloned out the genes for the alpha chain and the beta chain T cell receptor that was specific for H2KB. Now they've got this functionally rearranged alpha chain gene and beta chain gene that's specific for H2KB, and they introduce those genes into CBA mice to make a TCR transgenic mouse that all of its T cells have a T cell receptor specific for H2KB and can kill H2KB bearing cells. Now, it's important to note that this T cell receptor has exceptionally high affinity for H2KB. They demonstrated this biochemically, and it can have high affinity because this receptor, when it underwent negative selection in the mouse, it didn't encounter H2KB in the thymus, right? H2KB is not in the thymus of this mouse because H2KB is not native to this mouse. It's very important you understand that. I don't want to go any further unless you understand that. I'll ask the two biologists in the audience, Barry and Rusty. Did that make sense to you?
Say again.
Does that make sense to you? To us?
It's complicated. Yeah.
Inordinately complicated.
Yeah.
But I want you to understand this point. This mouse has a T cell receptor that's specific for H2KB because you introduced genes for a T cell receptor that is specific for H2KB. And those T cells develop normally in the thymus. They survive negative selection because the H2KB is not expressed in the thymus of the mouse, right?
Mhm.
Is that clear?
Okay. Mhm.
Okay. Now, what we did, we got these mice from France, and we have two other mice that the French group made these, we made these. These mice are also CBA mice, just like this guy up here, but I've made them different colors because now we are expressing H2KB in specific parts of the thymic epithelium. In the cow mouse, we have H2KB expressed in the thymic medulla, and in the KQ mouse, we have H2KB expressed in the thymic cortex. And these H2KB molecules in the thymic medulla will be available to T cells that are developing T cells that come up to this medullary cortical junction, okay? And then we did the following. We mated our cow mice to our BM3 mice to generate cow by BM3 mice, and mated our KQ mice to our BM3 mice to generate KQ by BM3 mice. And these two mice now have an interesting problem. They're expressing, their T cells express a high affinity receptor specific for H2KB, and they're expressing H2KB in the thymus. Okay? And the question is, what happens? So, again, as I said before, in the H2K BM3 mice, H2KB is expressed in the cortex. In the cow by BM3 mice, it's expressed in the medulla. We're the ones who showed that. And we believe, because of the biology that I'm going to report to you, that includes thymic epithelial cells that are at the medullary cortical junction. So the questions we want to answer are all the autoreactive thymocytes deleted in this model, or do some of them escape? And if they escape negative selection, what happens to them? Now, sneak preview, the mice do not get autoimmune disease. So whatever is happening to them, it's not, if autoreactive cells survive, they're not causing disease. And that's in itself very interesting. So I'm going to tell you how just a little bit of a little tiny nugget of our studies. And I'm going to tell you that because the BM3 T cell receptor is specific for H2KB, that means the T cells express CD8 because CD8 positive cells are specific for class 1 MHC, and H2KB is a class 1 MHC molecule. And if we fail to see the development of CD8 positive T cells, that indicates deletion of cells that are expressing this receptor. Before I go any further, I want to make sure that that makes sense. Please don't just sit there and suffer in silence. Say something, please, I beg you.
Okay, run over that again.
So, I told you, remember I told you, let me go back to this slide over here. I told you that CD8 positive, cytotoxic T cells have CD8 on their surface, which binds to class 1 MHC. So if a T cell is specific for class 1 MHC, it develops to express CD8 only. H2KB is the class 1 MHC molecule. So if a developing T cell that expresses the BM3 T cell receptor gets deleted by negative selection, you won't see these cells in the thymus because you won't have the development of these CD8 positive T cells. Okay? Did that help?
Mm-hmm. Okay.
All right. So, if those T cells survive positive selection, we will see CD8 positive T cells in the thymus. If they don't, that's because they died. during negative selection. Oh, I want to point some, well, okay. Now the problem is that all B cells and T cells look alike. So how can we study them? And for that, we use some very powerful tools. First of all, there are antibodies. Immunologists spent the 1960s and 1970s and 1980s generating a vast library of antibodies that are specific for all of the different molecules expressed by T cells and B cells. Really, the number is staggering. And also, it's possible to conjugate fluorophores to them, these fluorescent molecules. And so we have available antibodies specific for these molecules that we can conjugate to different fluorophores. A fluorophore is a molecule that if you shine light of a particular wavelength on it, it will emit light at a different wavelength. And there's a whole array of fluorophores that we use in our research. We have to select them so that their light emission is distinguishable. So for example, one fluorophore we use is called PAC blue. Another one is AF48 and so on. And you can see that we can, using an instrument I'm going to tell you about called a flow cytometer, we can see the cells that are binding a particular antibody. For example, our CD8 antibody is conjugated to PAC blue, Pacific blue, and it will give, if a cell is expressing CD8, will have emission of light at this particular wavelength. For CD4, we use various fluors. In this particular experiment, we used a fluor called AF48. It will emit light at this wavelength, and so on. So we are routinely using these fluorescently tagged antibodies with a very powerful instrument called a flow cytometer. This little diagram shows you how the flow cytometer works. You incubate your cells with a whole array of antibodies. We routinely use 8, which are conjugated to all of these different fluors. You run the cells through the flow cytometer in a single cell manner. So each cell goes through one at a time. We can run between 6,000 and 8,000 cells per second through the flow cytometer. And for each sample, we collect data on millions of cells. An argon ion laser will shine light on these. Well, we have several different lasers. Our instrument has 3 that will excite these different fluorophores and cause them to emit light at different wavelengths. And then through a series of optics and mirrors and various other things, the instrument will integrate all of these data for you and give you data that is both quantitative and qualitative on your cells. So for example, if I might have in my single cell suspension cells that express all of these molecules, the green, the red, the orange, etc. I might have some that only express the red and the green, or only the red and the orange. I can distinguish all of those cells. And this is important because we now know that different molecules on developing thymocytes are expressed at different times in their development. Okay? So we're no longer stuck with cells that all look the same. We can distinguish different cells and we can quantify them. Moreover, it's better than that. The data are displayed logarithmically. And that means that if one cell type has, say, 3,000 of these green molecules and another one has 50,000, I'll be able to tell what it has. And that's important because the developing T cells either begin to express or cease to express a molecule. And you can tell if they're in transition between one stage and another, depending on whether they're expressing a little bit or a lot of a particular molecule. Does that make sense?
Mhm.
Okay. So I want to just show you a little bit of our data, and I thought I'd start with baby steps. I'd come back to CD4 and CD8, which I've already told you about. So when the youngest thymocytes do not express CD4 or CD8, and they're referred to as double negative. Then as they rearrange their T cell receptor, they express both CD4 and CD8, and then they undergo scrutiny through positive selection. It's at that stage that they're expressing both CD4 and CD8. And then after positive selection and negative selection, if they survived both, they're either expressing CD4 only or they're expressing CD8 only. And these are the cells that go to your peripheral lymphoid organs. Okay? Everybody with me so far? So what we have here on the right is an example of a flow plot that we would get. So on the y-axis here, we have CD8 labeled with PAC blue. On the x-axis, we have CD4 labeled with AF48. You can see here that this is a logarithmic scale. I'm sorry that this is fuzzy, but here's 10 to the 0, 10 to the 2, 10 to the 3. Here, 0, 10 to the 2, 10 to the 3, 10 to the 4. So here are what we're referring to as our double negative lymphocyte, the T cells, those right here that are the youngest. Here are the double positives. They express both CD4 and CD8. Here are the ones that express CD8 only. And here are the ones that express CD4 only. All right? So these are the youngest cells. These are the ones that undergo positive and negative selection. These are the CD4s and the CD8s that survived both positive and negative selection, and that will go to peripheral lymphoid organs. And we can quantify. So here, these are 6.86%. These are 7.89%. These are 1.25. And this is 80. I can't read that because my, let's see, 82.5. All right. So now, what happens in our mice? Because the T cell receptor binds too well to the MHC, in the CalBiBM3s, we don't have that development of the CD8 single positives. See this wing? My lab refers to this as a bird. Here's the tail, the head, and the two wings. This CD8 positive wing is missing in the CalBiBM3s, but it's present in the parental BM3 T cell mice, which we expected, and it's also present in the KQ by BM3s. Okay, does everybody see that? I can no longer, I had to quit showing your pictures and I want to see them. How can I do that again? I don't want to stop sharing. I'll just have to grin and bear it. But the other thing that we noticed is that there's far more double negatives. So here the double negatives are around 1 to 2%, and here they're close to 16%. And we have gone on to study these double negatives, and we know that in the CalBiBM3, these cells are heterogeneous. They include both the truly immature double negatives that we talked about up here, but they also, using other markers of T cell activation, there's some that will let you know if the T cell encountered its antigen. We know that there's a significant fraction of what I refer to as antigen experienced thymocytes in here. And we've studied those further, and I'll just mention what we got in a minute. But I can't see your faces, so I want to know if you understand everything, and if not, please speak up.
Yeah.
I wish I could see your face. It's frustrating.
I think you can move on.
But if you didn't understand, I don't really want to move on.
Well, I guess what waiting now is an explanation of what this different production of the different cell types means. I guess
Okay.
yeah.
Okay. So our findings thus far, I thought I wouldn't burden you with any more flow cytometry graphs. Our findings have allowed us to determine the following. In the CalBiBM3 mice, but not in the KQ by BM3 mice, the selective force of having H2KB being expressed in the medulla has led to three things. As this first slide showed, a failure of the development of CD8 single positives. We know that because when we look in lymph nodes, We don't find any CD8 positive T cells at all. Secondly, there's a development of an unusual population of T cells that express the BM3 T cell receptor, but they don't express either CD4 or CD8, so they're activated double negatives. And we have found that these double negatives migrate to the lymph nodes where they express markers that are characteristic of what immunologists refer to as an exhausted state. They are unresponsive to antigen. This exhausted state of T cells is a fascinating one. We find it in T cells that in people who have cancer with a bad prognosis, they've got T cells, they've got CD8 positive T cells, but those whole things are all pooped out. They're not working, they're exhausted. And they express certain molecules that are immunosuppressive. And so we're pursuing these particular double negatives much more vigorously now. We're trying to study them better. We think they might be responsible for the fact that these Cal by BM3 mice don't get any autoimmune disease and aren't very good at rejecting Cal by BM3 skin grafts. And then finally, the third thing that we see is the development of these intraepithelial lymphocytes, or IELs, in the intestines of these mice. These IELs express the BM3 T cell receptor, and they also express immunosuppressive markers on their surfaces. And that's interesting because research on IELs and other systems indicates that these cells maintain tolerance to self in the gut as well. So the take-home message is that these T cells do leave. They don't stay in the thymus, they leave, but they're different. They're not normal CD8 positive T cells. They're double negative T cells that go to the lymph nodes and to the intestine, where they express these immunosuppressive molecules and are presumably unresponsive to H2KB and might inhibit autoimmune reactions. And what we'd like to know now is actually developing further elaborate other models that will allow us to study their function and also to see if we can reverse that lack of responsiveness. Again, if we think about cancer, it's bad for a patient. The prognosis is bad if you have T cells that have this immunosuppressive phenotype. You want them to be able to see the tumor antigens and to react. And so there's interest in trying to figure out how we can, do these exhausted cells, can they get a good night's sleep and no longer be exhausted, basically?
I have a question.
Yes.
If it's appropriate now. It sounds like this might be related in cancer patients to what they use checkpoint inhibitors
Yes.
for.
And in fact, actually, thank you for using that term. I'm going to share, now that I can see your faces again, I'm going to share my slides again. Yes. So these immunosuppressive molecules, one of them is called PD-1. And PD-1, the Nobel Prize for Physiology or Medicine was given for the discovery of PD-1, and it is now regarded as a checkpoint. Although I don't like, for PD-1, I don't like the use of the term checkpoint because it's not a checkpoint in the traditional sense. But anyway, antibodies against PD-1 are given to cancer patients to see if they can inhibit the action of PD-1 and can activate the T cells to be potent again in killing tumor cells. Yes, absolutely. Did that answer your question?
I think so, but I don't understand. I didn't get what the function of PD-1 is. You're saying
right.
It's not really a checkpoint.
Okay.
What is it exactly?
This is the way I'd like to explain it. Let's say you're having a party. Let's say on New Year's Eve, you're going to have a party. And after everybody has toasted, if they've stayed up past midnight, they toast each other, they drink their champagne, and then what do you want them to do? You want them to go home, right? You want everybody to go home. You want to be able to get to bed and start a new day. The immune system uses these so-called checkpoints to bring immune responses to a halt. You don't want an immune response to go on forever. You want the partiers to do what they're going to do and go home. And so PD-1 and another molecule similar to it called CTLA-4, which is considered a checkpoint inhibitor. The Nobel Prize was given for that too. Antibodies against CTLA-4 are also used to treat cancer patients. The function of PD-1 and CTLA-4 normally is to rein in immune responses. If you are mutant for PD-1 or CTLA-4, you can end up with lymphoproliferative disorders and diseases because you need those cells to go home. You need them to leave. You need them after they've done their job to be done with it. And CTLA-4 and PD-1 bring everything to a close. Unfortunately, in certain conditions, such as in cancer, it seems as if this constant emergence of cancer cells just becomes so overwhelming that the immune system says, okay, I'm done. And it starts to express these molecules that now halt the immune response at the very time that you don't want it halted, right? You want to keep getting rid of those tumor cells. Did that help at all?
Mm-hmm.
And now in our
Yes.
model, yeah, was somebody going to say something?
No.
I said yes,
Okay.
and it did help.
And in our model, what we're seeing is that the cells that go to the lymph nodes and to the IELs are expressing, in the CalBIBM3 mice, are expressing high levels of this PD-1. It's as if somehow during their development in Thymus, they were instructed to, okay, you survived, you get to go out, but now you're going to be a policeman. You're going to enforce tolerance to self. And you're doing it by expressing this immunosuppressive molecule.
Mm-hmm.
And one thing I really like about this is that it's characteristic of the immune system to the extent that it never has just a plan A for doing something. It has a plan A, a plan B, and a plan C. It really has various stopgap measures to make sure that it functions normally.
Hmm.
So-
I have a question. Am I right that autoimmune diseases are more common in females? And is this
Yes.
understood why?
It is understood to a certain extent why. It has to do with, well, I want to, before I answer that completely, I want to mention a few more things. This is true both in mice and in humans, and we know that estrogen is important. Some pioneering studies that were done back in the 70s, there's a strain of mice called NZB for New Zealand black, and another one called NCW for New Zealand white. And when you breed NCB to NCW mice, they end up with a huge propensity to develop autoimmunity, especially lupus. If you take an NCB by NCW male and you castrate him and you give him estrogen, he will get more aggressive autoimmune disease at a younger age. And if you take the female and you remove her ovaries and you give her testosterone, she will be protected longer from autoimmune disease. So we know that estrogen is involved. There's been some studies done in mice in the last several years that show that that estrogen potentiates immune responses. In fact, when we used to be making monoclonal antibodies in my lab, we always immunize female mice because you get a much better response from females. So at least in mice, females give you more robust responses in general. And the greater robustness of the immune response is probably why autoimmunity tends to be more aggressive in females than in males. Now, the part that we don't understand, since I told you that autoimmunity tends to develop later in life, both in humans and in mice, and has something to do with that thymic involution and a compromised negative selection, we don't know if estrogen's also playing a role in that maybe negative selection is more robust in males for reasons that we don't understand yet. But yeah, autoimmunity, it's not so great to be a female. But if you want a good immune response, better to be a female.
Well, maybe I'm wandering a little a ways here, but kind of related to that. I mean, females have to make sure they don't reject the fetus. And there's
well,
a
that's all done by
connection there.
well, that's all done by completely different mechanisms that enforce tolerance at the fetus mother interface. The specific kinds of cells that promote localized tolerance. So it's interesting that it is localized. A pregnant female is not in any way immunocompromised against foreign antigens. The tolerance is sort of specific to the placental maternal border. Yeah. I wanted to just, I still want to take questions, but I just wanted to finish with my very last slide. My lab is somewhat unique at UCSC to the extent that I run it exclusively with undergraduates. All of the research that I've reported to you was done with undergraduates. Here are 4 of them who are no longer here. Yitzhar is now in medical school. These 2, Lisha and Alana, are working in labs, research labs at UCSF. Shahar here has moved into, he's more interested in doing computer technology now. But in any event, I took this picture of them at the Midwinter Immunology Conference. I take my students every year to this international conference, which is held down at Asilomar. And this poster that presented some of their work, you'll recognize here some of those fax plots, won a prize for best poster. And this is significant because all of the other posters were presented by graduate students and postdocs. So I'm very proud of my undergraduates that they can be competitive in their research and in their presentations with graduate students and postdocs. I work side by side with them in the lab. I'm at the bench with them. We're very, very close. I tell them we don't have a Zuniga lab group, we have a Zuniga lab family. So
that's great.
Any
and
more questions?
How has it worked out during COVID? To what extent have you been able to keep things going?
Oh, you know, I was ready to weep. I thought that my lab was going to shut down because I normally get undergraduates into my lab through word of mouth. Undergraduates will meet other undergraduates in their classes and they'll say, oh, the Zuniga lab is a great place to work. You should come ask Professor Zuniga if you can work with her. And I thought, okay, that communication chain is severed. But actually, the opposite was the case. I was inundated with requests. What was challenging was, particularly during the hard shutdown when we could only have 1 person per 500 square feet. My lab is little, so that meant there could only be 1 person in the lab at a time. It was very, very hard for us to get anything done. But I decided to take advantage of that time by teaching them how to do all of this flow cytometry data analysis. And although the university can tell me how many people I can have in my lab, it can't tell me how many people I have in my house. And so we all wore masks and sat several feet apart. There were some out in the atrium, some in the living room, some in the dining room, and we did data analysis. And I didn't have everybody here at the same time. I'd usually have only 3 or 4 at a time. And so it actually turned out to be, okay, we lost a lot of time in the lab. I'm not going to say it was good for us, but you know, you have lemons make lemonade. I feel as if we became much closer. That's when that family feeling became even stronger. And it increased their commitment to the lab a lot because they could take their classes over Zoom here. So they could be sitting here at my dining room table and all of a sudden have an 11 a.m. class and say, okay, I'm going to go into another room because I have to go to my class. And then they could come back in here to work. And so it was much more, it's as if our lives were much more integrated with each other during that time. And they really got to see how the data analysis is done, and they could come to own the data in a different way than they did before. Because when we were meeting only in the lab, it was a little bit more challenging to get them to learn how to do the data analysis. It's really daunting at the beginning. Well, actually, not just at the beginning, it's always daunting. And they just didn't have the bandwidth to think about it. But now that we weren't doing experiments and we were mainly doing data analysis, they had the bandwidth to focus on it. So it turned out not to be so bad. I mean, we're still paying mightily. Our mice didn't survive the pandemic that well. The combination of the pandemic and the fire, which meant that we couldn't get up to campus to do the things we needed to do, we're still not completely recovered in terms of our mouse breeding.
Martha, I have a question.
Yes.
As a non-biology major, what are a couple highlights that you'd like us, like me, to go home with from your work and your presentation today?
Okay, thank you for asking that question. A couple of highlights. Well, I definitely want you to know that you're generating new T cells and B cells every day of your life, and that because they generate their receptors randomly, the other thing that's happening every day of your life is this elimination of cells that aren't good for you and the selection of cells that survive. And also, I sort of feel as if our findings about the double negatives in the lymph nodes and in the intestine, that's a really wonderful result. It's as if, okay, the immune system says, I can't necessarily delete every cell, but I can silence that cell. It's still alive, but now, it's now a government informer. It's now working for me, even though it would have been against me initially. Those are the things that I think I'd like you to take away.
Thank you.
Okay. Any other
Anybody
questions
else?
for Martha? Okay.
I have a question for you. I'd like some of you to tell me what was the most interesting thing you learned from me today, or the most fun thing you learned.
Okay, you can speak up.
This is a question I ask my students after every lecture. After every lecture, I ask them to tell me what's the most interesting thing or the most fun thing they learned.
I think the most fast. Oh, go ahead.
What? Oh, wasn't that it was this particular thing was interesting, but it fortified my understanding little by little of how incredible the human body is. It just blows my mind. Anyway, that's all. Just
Yeah, I agree.
supported that.
Yeah.
Ray?
Yeah, I'll say ditto to that. It's remarkable.
I was struck by the sort of analogy, I guess, between random mutations and natural selection in evolution, and the random generation of these sequences with a kind of a programmed selection in Thymus that it's a very similar process in terms of arriving at stuff that works is just sort of guessing, throwing stuff at the wall and see what sticks. And it seems like when you get to a certain level of complexity, the only way to solve it is some form of evolution and selection.
Yeah.
I don't know. It's just fascinating to me.
Yeah. I like your comment about throwing things at the wall to see if it sticks, because the immune system does that. We generate millions or perhaps hundreds of millions of T cells and B cells every day of our life. Most of them are going to die because they don't pass positive and negative selection. And then the ones that survive, you're hoping that some of them will be able to protect you against the new virus variant that appears. And if they don't work, it kind of doesn't matter because you're bringing out new ones all the time, right? It's sheer numbers.
So, yeah, so it makes me think about these variable regions that, we do all the mix and match thing. They're variable, and yet I assume it's not just random, right? So it must be some weird combination of a kind of fixed driven amount of different sequences, but it can't just tolerate completely random mutations. I mean, if you get mutations in those variable regions, are they selected against, selected for? The evolution of that gene is mind-boggling.
Well, okay, so Barry, I want to address that point. So you have the variable, the V region, D and J for the beta chain, and V and J for the alpha, and the variability in those loops that I pointed out to you occurs by bringing V to J or VD to J. The mutation occurs just at those junctions, just in those loops, not anywhere else. So you can have, let's say, variable region V beta 52. V beta 52 is going to stay, it's not going to mutate, but it's going to generate different antigen receptors depending on which D and J it's joined to and what mutation occurs at the junction of V with D and D with J. And those, of course, are somatic mutations. That's very, very important to remind. I didn't say that. It's not in the germline. It's only in that developing T cell, that mutation.
Mm-hmm.
That's important to bear in mind. The mutation is completely just in the developing T cell, not in the genes that are in the genome, right?
Yeah.
Now, I can't remember which of you, if it was Mark or you, Barry, who said that there must be some order. It's kind of interesting that for B cells, many, many years ago, a group in Georgia showed that in fetal life, which is when these VDJ and VJ joinings first start, they were looking at B cells because B cells also use variable regions and have VDJ and VJ for their immunoglobulin genes. And they showed that in fetal life, but not in adult life, you actually do selectively use certain Vs and Js. They never, it didn't really go very far in terms of explaining why that was the case. I mean, in fetal life in a mouse, they were looking in the mouse, the immune system isn't actually able to do anything yet. So what good does it do you to be doing these rearrangements? And is it practice? It's not at all clear. By the way, I should mention, not that anybody asked, but it's the mouse gestation is 20 days and the rearrangements start around day 13. So the last week of fetal development. Humans our adaptive immune system starts to generate functional T cells and B cells in the third trimester. And I don't think anybody's proven that a baby in the third trimester is mounting good immune responses, but it's fully possible that in utero, a baby could be mounting immune responses if a virus crossed the placenta or something.
So, well, maybe I'll ask what might be the last question here, going back to the COVID story.
Well,
So,
actually, let me make
Rusty.
I just have one little comment, Martha. I find it rather intriguing that sometimes evolution has selected the C student.
Yes. Right. Yeah.
Okay. So in the mRNA vaccines that most of us are taking, how many different variants, a kind of antigen recognition sites are likely to be generated? I mean, thinking of this with the current news that, you know, the spike protein antigen is mutated in this new variant. So I'm not articulating this very well, but we have mounted a host of antibodies, which is now facing a host of different antigens. And the bottom line of the question is, you as an immunologist, do you think, for example, this Omicron is going to be a problem or not?
I am hopeful that it's not going to be, for the following reason. The immune response, so when they titer, you know, they don't do it for you and me, but if you were in a clinical study, they would certainly do it. They would take your blood and then they would titer your antibody to see how much antibody is required to neutralize the virus, all right? And that scale, again, is logarithmic. So let's just say, for the purposes of argument, that if the vaccine that we got was against the alpha variant, our antibodies might bind the alpha variant with such high affinity that even if those antibodies are present at a low concentration, they'll be able to bind the virus with high affinity and take it out of commission, neutralize it. The booster in particular has, I understand, I haven't read the paper yet, but I saw the paper, that it seems that it's boosted, the first immunization got you this level of antibody. The second one maybe increased it about tenfold or something like that. The booster, apparently it's more than that. It's like 100-fold or 1,000-fold better. So everybody was surprised by that. They didn't expect it to potentiate the immune response so well. So the antigen binding sites of an antibody molecule consist of several different amino acids. Again, it's loops. I'm trying to look at things on my table to see if I can. So imagine that I'm holding this Kleenex, that if this is the alpha variant, that all 5 of these of my fingers are binding it really well with high affinity. And then with the Omicron variant, if there's certain mutations so that 1 or 2 of these fingers can't bind, you could still be binding with the others. And so you might need a higher concentration of antibody in the blood to neutralize that virus, but it's still, you can still have some affinity for it. And they're thinking that because the booster so vastly increased our antibody titers, that even if the Omicron variant can't be bound as well by the antibodies, because there's so much antibody around, it'll take it out of commission anyway.
Okay. All right. Maybe on that.
Did that answer your question, though? I'm not sure.
Well,
I was.
It helps a lot. Yeah, it helps a lot. Yeah. And as I said, on that very positive note, maybe we'll bring this to a close. So thank you very much, Martha.
Thank you. Thank you, everybody. This
Hey.
was fun. I always enjoy getting together with you very much.
All right. In the future, in person too.
Yeah, and in the future, in person.
Yeah. Okay. And also, you know, Lois Whittam is on here, and she's in charge of our courses. And Lois and I have, you know, want to plant this little idea in your mind of, you know, you could do a whole course for us.
I could do a whole course for you.
No, please.
And as you know, Barry, I really love to teach and to explain things. So, yeah.
Okay.
I hope you all got something out of this. I hope you're not bored or so confused.
Not bored, Martha. No.
Really very interesting.
Okay. All right. Well, thank you, everybody.
Confused but
Thank
not
you.
bored.
Okay.
Right, there, that's better, right?
Okay. Well, next get together will be the 3rd Sunday in January, when we will have an OLLI talk completely different. It'll be Shakespeare and the lessons of Hamlet for our modern days. All right.
Wow, that
Okay.
sounds interesting.
Okay. See you, everybody. Okay. bye.
Okay, thank you. Bye.
Bye. Bye. Bye all.
Okay, where are we doing here? Down here.
Thank you again, Barry.
Okay, well,
Happy
thanks a lot,
holidays, everybody.
Martha. Okay.
Martha, you could join OLLI. Nice organization.
Yeah, yeah.
Yeah, I could.
Yeah.
Anyway, I hope that wasn't too overwhelming. Was it too overwhelming?
Well, it's a really complicated story, so it's hard to, to get all the pieces in your head. But I think, we got the drift of it. Yeah. But I've been looking at this a lot. I've always been puzzled by the immune system. It's just so complex. And it helped me today because I had never quite gotten that DJ joining thing. All right. Okay.
Well, I want you to know that immunologists find it overwhelming and confusing too. And I still have this feeling of you've got to be kidding me that this is the way we do things.
Yeah.
Yeah. That's
Yeah. Right.
It is mind-boggling that it has survived. I really think it's this numbers game, right? If you're generating hundreds of millions of cells every day, then you don't really care if only a tiny fraction of them can do the job.
Mm-hmm.
Profoundly wasteful.
Yeah.
Yeah. Yeah. Who attacks the non-functional cells? Macrophages? Or
Yes. Macrophages in the thymus eat them up. They die by apoptosis and then they get taken up by a specific kind of macrophage called a tingible body macrophage. Yeah.
Okay, but the signal is the apoptosis.
Yes, right.
Yeah.
Yeah.
Mm.
Anyway,
All right. Well, thanks everybody.
thank you. Bye.
Bye.